Katie is assistant professor in plant microbial biology at University of Minnesota, Twin Cities. Before forming her own group, she was awarded NSF Plant Genome Postdoc Fellowship at Dartmouth College, where she studied how polyploidy and gene expression regulate and underlie stress responses. In brassicas, brassicas for non-plant folks are in a very wide collection of plants, but they also include cabbages. That's like bok choy, broccoli, cauliflower, cabbage, kohlrabi, Napa cabbage, rutabaga, turnip. So like half the produce aisle in the grocery store is a brassica. So they're important for other reasons other than being really interesting model plants. Yeah, I had some, oh, anyway, we'll skip that. Her group at the University of Minnesota studies plant circadian diurnal rhythms, specifically testing genetic basis of complex interactions between internal timekeeping mechanisms and plant responses to environmental stimuli. She's also served on the JGI User Executive Committee since 2020. So please welcome Katie to tell us about brassicas, or her favorite brassicas anyway.
Thank you, Jeremy, for the introduction and the invitation. Yes, so I'm going to talk to you today about brassicas. I don't now need to tell you why they're so amazing, because Jeremy's done an excellent job of that. I'm glad he didn't continue on with the story, which I think was going to take a turn for the worse, maybe. So today, mostly I'm going to talk to you about things on the daily scale, but I think it's important to remember that changes in our environment, photoperiod and temperature, which are driven by the rotation of the Earth, vary by latitude. And so here's an example of two latitudinal zones, and I'm showing you across the year. In the solid line is temperature, and that hash line is photoperiod. So of course, photoperiod is fixed at that latitudinal zone, but what we see is that there's this lag in temperature behind photoperiod. So the warmest time of the year actually comes after the longest day of the year. Plants are accustomed to this relationship and are adapted to it. Unfortunately, climate change is skewing this a little bit. So the temperature now is shifting and that lag is becoming less predominant. So it's important for us to understand how the plant's perceiving these different signals if we're going to try to help adapt crops, especially to their environmental conditions. Similarly, though, on the daily scale, we also see this lag with temperature and light. So here again, those two latitudinal zones, the solid is still temperature. Now the hash line is solar radiation. And you can see now that the warmest part of the day actually comes after the brightest part of the day. And this is important because this conflicts with most controlled environmental experiments that we do, where light and temperature are usually synchronized. So lights come on, temperature goes up, and the same happens at night. And so really, again, even on a daily scale, this lag is shifting where we're seeing less of a change in temperature throughout the day. And it's becoming warmer earlier in the day. So it's really critical that we understand how plants are perceiving this change, because a change in temperature can trigger things like flowering even before it reaches that photoperiod induction. Now, how do plants sense changes in photoperiod and temperature? This is due to this internal circadian oscillator. And this internal timekeeping mechanism helps to coordinate changes in external environments with internal processes. So a great example is a change in photoperiod can trigger flowering time, but also things like responding to abiotic and biotic stress, metabolite cycling throughout the day. And ultimately, this helps to maximize energy use efficiency so the plant can maintain growth and fitness under a changing environment. At the core of the clock, which is conserved across all domains of life, is this regulation of the transcriptome. And this is shown here in a beautiful study in 2019 by Ferrari and colleagues, just showing the conservation of time of day control over the transcriptome. And what I want to emphasize here, this is across the Archaeoplastida. So you can see that the percentages above the heat map of the percent of genes in the transcriptome that show time of day expression. And you can see that it increases in complexity as you move from the prokaryote on the left, where you see it's a very on-off pattern. So genes are either on during the day or off. And then you move to the right to rice and Arabidopsis. You see this increased fine-tuning or complexity where now we have genes that are only on in the morning or midday or late in the day. So you can see here now that it's become much more complicated, but also this kind of sets the stage for many more processes being controlled to specific times of the day. In crops, the clock has also played a really crucial role in domestication. This is in large part when human populations migrated to different latitudinal zones and they brought their crops with them that now had to grow under different photoperiod and temperature conditions. And so here's just a few examples of some crops that we've now identified the traits that have been selected for during domestication, and the genes underlying those traits are circadian clock genes. So this is some examples in soybean, barley, and tomato, where we've actually determined that we've domesticated these crops and made alterations to their clock compared to their wild ancestors. And a lot of this has to do with being able to grow them at different latitudes. So this tells me that the clock is an excellent target for crop breeding, and really any trait of interest, we should be keeping in mind the endogenous clock in our target crop versus a wild if we're trying to integrate traits from the wild, and really thinking about how those traits are integrated into the clock network to be able to maintain that optimized growth. So again, since my lab's interested in time of day control, we think in terms of patterns. And this is at the level of transcription, physiology, and metabolism. Today, I'm going to talk primarily about the transcriptional control, but we're also very interested in the clock regulation of metabolites. So on the right here is actually an example from one of our JGI data sets looking at glucosinolates, which are the sulfur-rich specialized metabolites that Patrick Shih introduced us to yesterday. And these are made only in the Brassicaceae, so the mustard family. And we can see here that they also cycle throughout the day and are clock regulated. And we're seeing some interesting patterns with them in terms of their response to stress. So I'll touch on that a little later in the talk. So really the goal then is for any trait of interest, we want to identify the patterns that are associated with that trait. So for example, what are the patterns associated with this tolerant plant that's tolerant to an abiotic stress versus a sensitive one? And then if we see a certain time of day control associated with that trait, how do we move that time of day control into the more sensitive variety? So rather than simply overexpressing a gene to confer tolerance, which often has a yield cost, can we maybe fine tune the timing of when things happen to improve tolerance without knocking down all the other growth processes? So that's the goal. And here's how we go about that. So our system is Brassica, and there's a number of reasons. Jeremy also already alluded to why I love Brassica. First and foremost, it's closely related to Arabidopsis. So for a dicot system, that's really a benefit because we can translate what we know in Arabidopsis into a model croc system. We can take Brassica genes and test their function in Arabidopsis quite easily. It's an excellent model for studying polyploidy because of its recent triplication following the divergence from Arabidopsis, where every gene could potentially have up to 3 copies. It's also a really nice model for Brassica napus or canola, which is a really important oilseed crop. We also do a lot of work in napus, which I'm not going to have time to talk about today. But just because of this incredible morphological diversity, it's a really good model system for other crops, and we can do it all within one species. So primarily what we're working on is Brassica rapa. So napus is an allopolyploid between Oleracea and rapa. So I'm really interested in how polyploidy and genome duplication has influenced the clock network. And what we know about the clock is that it has expanded throughout the green lineage. So going from a simple 2-component system in Austriococcus to a much more complex oscillator in Arabidopsis. So universal to clock networks across organisms, one of the trademarks are these negative feedback loops. And so here I'm showing you a very simplified version of the clock. There's a lot more members that we've discovered now, where you have morning or daytime expressed genes that are shown in yellow that tend to repress the evening expressed genes and vice versa. Now you can see as you go from Arabidopsis to Brassica, there's been an expansion in copy number. And what we have found is that circadian clock genes have been preferentially retained in multiple copies in Brassica, suggesting that they're doing something. They've maybe acquired new functions, they're diversifying in function, and maybe they're contributing to some of this morphological diversity. So when I started my postdoc at Dartmouth, that was one of my first questions, are what are all these genes doing? And I set out to do kind of a classic RNA-seq circadian expression experiment where plants were grown under light dark conditions to kind of entrain the clock to that condition. And then we remove all external stimuli and we shift them into constant light and temperature. So now any genes that are rhythmic are being driven solely by the internal circadian oscillator because we've removed any cues from the environment. So I sampled leaf tissue every 2 hours for 2 days and then looked at what genes were rhythmic. And so what we found was that over 70% of expressed genes were clock controlled, which is a pretty staggering number and much more than we've seen in Arabidopsis. So this is just showing you a heat map of the genes with their expression throughout the day. So in general, what you'll see on the top, those white bars are the daytime, and then the gray bars are the nighttime genes. And you can just see these waveforms of expression throughout the day, similar to that earlier paper I showed you. So what we also found is that genes that were retained in multiple copies tended to be expressed early to midday over the evening expressed genes, which was really interesting to us and kind of got us thinking about, well, have these genes themselves maintain similar expression patterns, or are they diverging in expression? So is there any indication that maybe they're taking on some new functions? So to do this, we had the first challenge was how do we really identify differential patterns in gene expression? So rather than comparing single time point expression differences, we wanted to know whether a waveform of expression was altered under different conditions or between 2 copies, for example. So we developed this package called DIPALM, which is an available R package described in this paper. So I'm not going to go into too much detail, but it is going to come up throughout the talk. So briefly, what DIPALM does is it takes advantage of a gene co-expression network where genes are clustered into these modules based on their expression pattern. And this does a really nice job of pulling out genes with specific phases or time of day expression. So we can then pull out the principal component or the eigengene for that module and then associate every gene's expression profile to that eigengene. And that essentially gives us a value that's representative of the pattern. Once we have that value, we can then feed it into any standard linear model contrasts and test changes in expression pattern. We can then cluster those genes to pull out genes with similar pattern changes. So getting back to that question of the paralogs then from this circadian data set, if we pulled out all the genes that are in multiple copies that were expressed in our data, we run them through DIPALM and we ask how many paralogs change in expression. We found that over 40% of them had different expression patterns, which is pretty striking given that this, RAPA hadn't diverged from Arabidopsis that long ago. So here's just 2 examples, so I'm telling you the truth. Here are 2 clusters, one on the left that has around 90 gene pairs, where one copy is expressed in blue here at the beginning of the day, and then the other copy is almost completely anti-phase in expression. And then on the right is another example where we see a phase shift of a few hours. So not only are these paralog pairs changing expression, but they're doing so as clusters or groups of genes, suggesting some regulatory structure. Okay, so this is cool. They're changing in expression. Is there any indication that they're changing in function? So for example, if we have a gene in Arabidopsis that we know responds to abiotic stress, and there's 2 copies in RAPA, are they both responding, or do we see that one is responding and the other one isn't? And that would be a really great target, for example, if we can alter one for stress tolerance, where the other one maintains its other functions. So those are genes that we'd be interested in. So this brings me to this JGI CSP that I was awarded near the end of my postdoc, a project which a data set I could never possibly generate without the resources of JGI and Hudson Alpha. This essentially generated 7 de novo genome assemblies for different morphotypes in RAPA, which are almost complete now, which is really exciting. In addition, a massive RNA-seq and metabolite data set where I took 15 accessions that covered these morphotypes and ran them through a cold stress experiment. So here you're seeing the control conditions on the top, cold on the bottom. So these were in cold stress conditions for 3 days. This is more of like an acclimation condition to simulate sort of a late, leading up to a late frost in the spring. And on the last day, leaf tissue was collected every 4 hours during the night and the day, and then a few time points in the recovery. So we collected measurements of photosystem 2 efficiency throughout the time course, and then also RNA-seq and metabolite sampling to capture those glucosinolates that I mentioned before. Now, since we're still working through this data set, I just want to focus in on 3 varieties of this yellow sarsen oilseed morphotype. 3 genotypes that we ran, ACC28, ACC50, and R500. This is work done by a former postdoc in the lab, Matti Oravik, who set out to first assess the tolerance of these lines. Because if we're going to associate changes in transcriptional pattern with stress response, we need to classify their tolerances. So here's the design that Matti came up with, and I just want to highlight here that we've now kind of staggered our cold treatment to mimic a more natural environment. So the cold, that freeze that these plants experience will happen at the end of the night and into the morning hours. So for these 3 varieties, it was pretty clear after we repeated these experiments that ACC28 was the front runner. It seemed to tolerate this freeze much better than the other 2, whereas R500 got hit pretty hard, as you can see in these pictures. And this is just quantified here by leaf number following the stress. You can see ACC28 does pretty well under the freeze. Oops. And then ACC58, not so well, and R500 even worse. And then on the right are just the damage ratings for the damage scores of the leaves following the stress. And then we also look at photosystem 2 efficiency, which is very dynamic throughout the day. So we'll collect numerous time points of that. This is one example of a time point 7 hours after dawn. And you can see here, this fits nicely with that phenotyping data where the ACC28 drops its photosystem 2 efficiency in the freeze, but then recovers really quickly once it returns, unlike the other 2 genotypes that don't do as well, and R500 really struggles to come back. Now, this is for the oilseed varieties. I should mention for other morphotypes like Chinese cabbage, in a lot of cases, we see an increase of photosystem 2 efficiency in the freeze. So depending on the morphotype we look at, the response is very different. And this is something we're really interested at looking at the transcriptional level, because it's going to help us tease apart, what specific pathways these morphotypes are using, because there's many different ways of being tolerant to a stress. And so it's easy to forget that. And that's something we're excited to look into in this data. Okay, so we've classified these 3 morphotypes, these 3 accessions, excuse me. So now the question is, what are the transcriptional patterns that are driving these different phenotypes? So the work I'm going to talk about now, actually the rest of the talk is primarily by this graduate student in the lab, Angie Racono, who was assisted by this former undergraduate in the lab, Eileen Casola. So just to remind you, again, these samples were taken every 4 hours on that 3rd day of the cold stress, in the night and during the day. So we ran them through our pipeline and asked how many genes are now changing pattern in response to cold. And the numbers were enormous. So on average, there's about 24,000 expressed genes in our data set. A good portion of them were changing. But if you think about it, anytime you do a single time point analysis, you can easily generate a couple thousand genes. We're now looking at 6 time points, and we're kind of seeing that full set of dynamics throughout the day. But it's really also suggesting that really there's a lot of alterations to the transcriptome as a whole in response to this cold. So again, just to show you some examples, these are the top 2 clusters for each of those genotypes. These are now normalized, so we can just look at their patterns. So in blue is the cold, in red is the warm, the control conditions. And you can see here, these are covers about 700 to 900 genes for each genotypes that are showing similar changes in response to cold. But what I hope you also notice is that a lot of these are changes in phase, so the timing of peak of expression. So these are specific time of day changes in response to cold. So we wondered then if any of these genotypes have a specific time of day where we're seeing a lot of things happening. So to get at that, we took advantage of this well-known circadian program called JTK Cycle, which analyzes time course expression data and identifies genes that have rhythmic expression and assigns a phase value to them. So we could do this for all our genes, pull out the rhythmic genes, and identify their phase under control and cold conditions, and then ask how much did their phase change in response to cold. So here's the distribution for those 3 genotypes. So the purple arrows indicating genes that have no phase change. So they might just simply be changing their median expression level or an amplitude change. Whereas anything to the right of the arrow, every bar is a 2-hour increment. So that would be a delay. So the gene's now peaking 2 hours later. Anything to the left of the arrow would be an advance. So the gene's peaking earlier. So what immediately jumped out to us, and hopefully to you as well, is that ACC28 on the left, which is our more tolerant line, has a shift in this distribution where there's a lot of genes that are delayed in their expression by 2 to 8 hours. Whereas the other two genotypes are more normally distributed around that 0-hour change. So this was really interesting. And we wondered, of course, what are these genes in these categories? So what do you do when you have a bunch of genes? You run them through Go enrichment and you look to see what they're involved in. So here's just a few examples of those. We have a lot in each of those phase change groups. And so here I've just arranged them with bars to remind you of what phase group they're in, and then color coded by the genotype. So a few that are really interesting to us are these glucosinolates, which we found in the really tolerant, and then the ACC50, which was kind of the mid-tolerant line. So there's this huge shift in genes that are involved in glucosinolate biosynthesis and metabolism. And I told you we have this metabolite data set that we're mining now for glucosinolates, and where we see changes in their abundance in response to cold. And now we're also seeing that at the transcriptional level as well. So we're really interested in trying to understand why glucosinolates would be increasing under these abiotic stress conditions and what they might be doing. But that response seems to be completely absent in this R500 line. So we have now a bunch of categories, and we have a bunch now that have specific time of day responses. But before I get back to this, I want to get back to that original question about the paralogs. I showed you earlier in the circadian data set that we had a lot of paralogs that were changing an expression pattern, and we wondered if any of them have certain, if they've specified in their ability to respond to abiotic stress. So is there any diversification in stress response? So now with this data set, we have the ability to answer that question. So again, with our 3 genotypes, if we pull out those genes that are in multiple copies, and we run them through DIPOM, under control conditions, now we see that over 60% of these multi-copy genes have different expression patterns. And then if we ask, of those genes that have different patterns, is there any evidence in divergence of stress response? And of course, I wouldn't tell you if the answer wasn't yes, we do. We see significant enrichment where one of those copies responds to the stress and the other one doesn't, which indicates that they may be then diversifying in function. What's even more interesting is that, or maybe more challenging for us, is that there's a tremendous amount of interspecific variation. So that paralog that responds is not conserved even within this one morphotype. So the overlap is pretty low. We're just over 70 of those genes. So when we do so when we look across these genotypes within a morphotype, the one paralog that responds is not the same. And so this makes it a little challenging for a breeding target perspective, and suggests to us, which a lot of us are already thinking about, is that regulatory elements are where the action is. And this is going to come up again in my last few slides. Okay, so here's just a few examples of some genes in those 3 genotypes that are known to be involved in cold stress. COR28 is also integrates cold in the circadian clock. The best way to compare it is if you look at the troughs, the low part of expression, and you follow it down, you can see that ACC28 does shift in terms of where its trough is, which is consistent with that kind of phase change we're seeing in ACC28. And then here's 2 copies of a transcription factor, BHL68. And you can see their expression in some is unique in ACC28, but also just within a genotype, the 2 paralogs have different expression patterns, which is what I've been mentioning with the different expression among these pairs. Okay, so in the last few slides, I'm kind of going to tell you where we're going with this data. It's great to have all these massive lists of genes changing expression, but if we want to alter their expression, we need to get to the transcription factors or the regulators that are driving this. So we're going to be taking a number of approaches. One are gene regulatory networks. So we're using a lot of different programs to do this. One example is Genie3, which when given a set of transcription factor will predict targets based on their expression pattern. So these assume both a positive and negative interaction. So here's just an example on the left. So essentially what we do is we'll generate these gene regulatory networks for each genotype, cold and control. And then we'll ask, what transcription factors have targets that are enriched for cold responsive genes? And again, since most of our genes are cold responsive, it's a very long list. So here are the number of cold responsive genes, and then each line is a transcription factor. And then if we ask, of these transcription factors, how many themselves are cold responsive, because we want to be able to alter a transcription factor that's responding to cold itself. And there's a large set within each genotype as well. So this is kind of our first line of filtering. And then because we want to target transcription factors, again, that are altering that cold network, we'll look at our gene regulatory network in the control data set and then compare those transcription factors. So if we look at our control network and then we run the same analysis, here's just in the mustard color, the transcription factors in our control network that are regulating those cold responsive genes. And then in blue is the cold. And when you look at the overlap, it is very small. So under the control network, those cold responsive genes are being regulated by other transcription factors. And under cold, there's a very complete rewiring where a new set of transcription factors are targeting these genes. So moving forward with this, now we're going back to those phase change groups to ask what transcription factors are regulating those genes so we can target a specific pathway like the glucosinolates. And then once we identify that pattern for that transcription factor, we'll then try to take a promoter element, move it into our sensitive line to alter that expression pattern. And lastly, just to kind of drive home this point, when we look at those transcription factors across those genotypes, the overlap is very low. So again, there are different paralogs that are responding. If instead of looking at the brassica genes, though, we convert them to their Arabidopsis ortholog, the numbers get a little bit better, suggesting in a lot of cases, the regulator is the same ortholog, it's just the copy itself that is different within the different genotypes. Okay, so just a few takeaways, because I'm definitely, I think, out of time. So hopefully I've convinced you that by classifying these temporal changes in expression, we can really capture the dynamics that we can associate better with metabolites and physiology that we know are very dynamic throughout the day. And then I've also showed you that cold stress is really eliciting these large scale changes in the transcriptome. And we're seeing some very interesting time of day shifts that are unique to certain genotypes. So now we're kind of following up to see if the clock itself is being altered in response to cold, and that might be driving a lot of this. And then finally, there's a lot of interspecific variation. And so finding a certain gene to target is going to be very difficult. But if we can we can take advantage of this, I think, and help to identify the regulatory regions that are conserved among the paralogs that are responding, and maybe use those to pop those into our sensitive line to alter an expression pattern. And with that, I've kind of highlighted the people along the way involved in the work. I want to, of course, thank JGI for their tremendous support, NSF, University of Minnesota, and you for your attention. And I'm happy to take questions.
Do we have time for questions for Katie? One. Yeah, one. Yeah, we're doing it. Anybody have questions, Katie? Great.
Hey, thank you. Great talk. I was just wondering if you could combine a little bit from the last speaker and maybe paint some of your open chromatin to label maybe more granularly, and what you thought about that.
Yeah, we would love to get some ATAC-seq data, and that's kind of, I think, our next step. We're just trying to figure out which genotype to do it in. There aren't many ATAC-seq time courses. So one question too is how dynamic it is. I have a feeling that maybe throughout the day, it's not changing very much, but maybe in response to cold, we can see some rearrangement. So that's definitely the next thing, is picking a few time points under the control and cold condition. And that will certainly help us kind of resolve our gene regulatory network a little better and refine the space for regulatory elements. It's a great question. Thanks.